classify smooth muscle based upon how it behaves, how it is innervated, and how it behaves. And on that basis, we can think about two types of smooth muscle units. Single unit smooth muscle, which is where a single neuron is engaging with all the muscle cells here. The muscle cells are also well connected by gap junctions. So notice how they're very much interconnected. And in this example, the muscle is acting as a single unit. They're going to be more synchronized contractions. And we're going to see this in areas of the body where we need to have an all or nothing contraction. So here, there's one autonomic neural end plate or autonomic neuron that is innervating this entire bundle of muscle that's acting as a single unit. Comparatively, we have the multi-unit smooth muscle. And multi-unit smooth muscle has neurons that are going all throughout the muscle to engage with each individual cell separately. We don't have the extensive gap junctions that are connecting the muscle. And so we're going to see this in parts of the body where we want a more customized contraction that's unique to specific areas. So this muscle is not acting as a single unit. Rather, each cell is sort of behaving as its own individual unit, and it's being innervated separately and will contract and relax independent of the other cells. So the two types of muscle, single unit and then multi-unit. Now, single unit is the most abundant type of smooth muscle. This is the most common feature. It's going to be seen in areas like the gastrointestinal tract, most of our blood vessels, and most of the respiratory tract. We're going to see that the fibers, again, connected by lots of gap junctions and then contract together as an individual, as an entire collective unit. Now, we can sort of make this connection. When we think about blood vessels, especially the smaller branches of the blood vessels, these cells and these structures need to act or contract synchronously. So when your capillary beds, when your arteries are contracting, or when the smooth muscle is moving, even in the gastrointestinal tract, this does not need to be as customized in individual sections. This needs to be entire collective contraction of the unit. Single unit muscle also is seen to have pacemaker potential in that this muscle can spontaneously depolarize. So in the absence of any neural stimulation, in the absence of any neurotransmitter leaving the neuron, this muscle can also just spontaneously manipulate its channels, its voltage-gated channels, and spontaneously result in depolarization. We're also going to see the innervation to few cells. So only the top layer here will receive the innervation, and then it will pass the signal on through the gap junctions so that the deeper cells can be innervated. And we're going to see increases and decreases in tension. So collectively, the muscle will either contract, which is increase in tension, or relax, which is to decrease that tension. Another feature that's seen with single unit smooth muscle, and especially seen in blood vessels, is tone. There is a natural contractile state of these muscles, which is called that natural tone. And that is a result of the pacemaker potential. Because the muscles can maintain a slight contraction, even when they're not being innervated, that is what keeps our blood vessels in that natural tone and keeps them slightly contracted in order for blood to move freely through, or in the case of the GI tract, or the luminal contents to move freely through as well. Another feature here, stretch reflex. So when we slightly stretch these muscles, right? When we slightly stretch a blood vessel, then that can lead to relaxation. And that is sort of a protective reflex or protective mechanism to prevent the overdistension of the GI tract, the overdistension of our blood vessels and capillaries. So a slight stretch results in relaxation, especially when that stretch is sudden. So it kind of automatically stretches beyond its natural tone or natural resting state. That results in automatic relaxation. Now, with the contractions here, we don't see recruitment, and there's going to be a varying amount of intracellular calcium that results in a varying level of contraction versus relaxation. And then the reason we don't see recruitment here is because everything is already contracting. All of these muscles are already beating as one unit, and so there is no additional units to call on or to recruit because they're all behaving as a synchronized unit. Now contrast that to multi-unit smooth muscle. This we're going to see in the large parts of the airway. We're going to see this in large elastic arteries like the aorta. The eyes, muscles of the eyes, such as the ciliary muscle and the iris, where we have that smooth muscle. So this is going to be areas where we need a more customized, a more specific type of contraction where the entire unit is not beating at one, but there are individualized contractions for each cell. There are very few gap junctions here, if at all. And then these fibers act individually. So each individual muscle cell receives its own innervation, receives its own neurotransmitter being released, and is going to interact with the neurons separately. Okay, we don't see tone here, and we do see recruitment. Because we have the individualized innervation and the individualized contraction, we can actually recruit muscle cells that are not yet being stimulated and add that in order to increase the tension production. So multi-unit smooth muscle does exhibit recruitment. Okay, and then I've got, again, another sort of simplified schematic that shows the examples. So up here, we're looking at our single unit smooth muscle, which has extensive gap junctions and a single postganglionic neuron. Remember, we're on the autonomic nervous system, so this is going to be a postganglionic neuron as opposed to a motor neuron, which would be innervating skeletal muscle. So this junction here is called the neuroeffector junction, and one of the features of that are the varicosities, the swellings that have the neurotransmitter. So the features we saw: small blood vessels, the gastrointestinal tract, and tissue that functions synchronously or as a single unit. Another feature here was tone, which is constriction in the absence of any neural input, and that is described as pacemaker potential. And then we don't see the ability to recruit here because no cells are not contracting, right? They're all contracting as one. And then compare that to multi-unit smooth muscle, no contractions, or no gap junctions rather, no gap junctions, no tone, because they're all beating individually and they don't have pacemaker potential. They can exhibit recruitment. And then the examples here are going to be large muscles that we want to have customized contractions. So large blood vessels, large parts of the airway, and structures of the eyes. Okay, now let's describe this pacemaker potential a little bit more and what it really means. This is the ability for the muscle to spontaneously depolarize. And when we look at the example of the action potential, we see that in order for the membrane potential to increase and for that to result in the stroke or the upstroke that results in action potential, we have to open up certain voltage-gated channels in order to do that. Now, with a pacemaker potential, those channels are spontaneously opened up, and there's a spontaneous depolarization that isn't triggered by a neuron or any release of neurotransmitter. So this is called a pacemaker potential, and there can be two different types of pacemaker potentials. So we can think about a slow wave potential, which is really an oscillating change in the membrane potential. This is also spontaneously triggered. So the resting membrane potential is not stable, but it's kind of oscillating because of the spontaneous opening and closing of the relevant voltage-gated channels. And then another type of pacemaker potential that we see is where there is actual spontaneous depolarization that results in an upstroke in the membrane potential, followed by a repolarization, and then that comes back down to rest. So these pacemaker potentials are much more sustained. They're much more meaningful in terms of resulting in an actual action potential. Whereas the slow wave potentials just make it easier, make it more favorable because of this oscillating change in the resting membrane potential, such that we could easier result in a full depolarization and a full action potential. Now, this pacemaker potential is going to be seen in single unit smooth muscle as well as cardiac muscle. So here we're going to begin to look at features that are seen in different categories of muscle, right? And recruitment, for example, is seen in skeletal muscle as well as multi-unit smooth muscle. So there are certain features that are shared across single unit and cardiac, as well as shared across multi-unit and skeletal muscle, right? Especially the pacemaker potential only being seen in cardiac and single unit. So moving on to look at cardiac muscle. So now that we've talked about skeletal muscle and we've talked about smooth muscle, we want to end off this chapter by segueing into what are the features that are unique to cardiac muscle when we compare them to the other two types that we have described. So cardiac muscle is very similar to skeletal muscle in that there's the arrangement of the actin and myosin into sarcomeres, which gives the muscle the striated appearance. We also see troponin and tropomyosin, which are the regulatory proteins here. Remember, we did not see these two proteins in smooth muscle. Instead, we still see actin and myosin, but they are regulated by calmodulin and MLCK. So troponin and tropomyosin are exclusive to skeletal muscle and to cardiac muscle. We're going to see gap junctions, and gap junctions is a feature that's shared with single unit smooth muscle. Again, notice the comparisons between single unit and cardiac. There's a lot of similarities across both of those. We're going to see pacemaker potentials. We're going to see the innervation by the autonomic nervous system. And then the calcium here is also going to come from the extracellular fluid and the SR, the so-called calcium-induced calcium release, is going to be a feature that we see in both smooth and cardiac muscle as well. The action potential will last for a much longer duration and will produce more tension than we see with skeletal muscle. And then summation due to the refractory period is not a feature that we see here. Summation is only seen in skeletal muscle. This is where we can add the tension because the action potentials are being produced with such frequency that they're really being added together because they're so close in time and space. So that is not seen with cardiac muscle, okay? In fact, the refractory period of cardiac muscle is so much longer that it's impossible for another action potential to come along and to be added to the existing action potential. The refractory period is too long for us to see summation with cardiac muscle. So here is the membrane potential, and here is the upstroke of the membrane potential, and then notice how long the refractory period is. So the refractory period lasts the entire duration of the repolarization period, such that when one contraction ends, within that period, it's impossible to stimulate the muscle again to result in a subsequent contraction. So it is impossible to have summation because the contractions are just much longer. All right, we're going to end off here by looking at a summary table. This shows the features that are shared across all four types of muscle, so skeletal, cardiac, and then both single unit and multi-unit smooth muscle. This is going to be a great way to study this and just try to fill this in from your understanding of each of these. Mainly the source of calcium. This is extremely important, how it differs for smooth muscle and cardiac muscle. And then other features such as the recruitment, only in skeletal and multi-unit. And then looking at the overall structures. So the striations are only in the skeletal muscle and cardiac muscle. And the way that this is innervated will be different, voluntary for skeletal, involuntary for the other types. However, notice that the actin and myosin, the thin and thick filaments, are present in all four of these types. The way they're arranged is different, the way they're regulated is different, but they're seen in all four types.